Okay, so this is a diagram showing the basic idea of RNA sequencing. If you haven't analyzed data before, this may be useful. So the object we're interested in studying is the full-length mRNA, ideally. But the technology, at least the next generation sequencing, or some people call this the second generation sequencing, that has been widely used for 10 years, has to sequence fragments of mRNA instead of the full length due to technology limitations. So after you fragment the RNA, there are steps that does reverse transcription, adding sequence adapter, and then PCR amplification. So those steps try to stabilize the signal and amplify the signal so they can be detected. There's often also a fragment size selection step to remove the too short and too long fragments and only keep the ones with a good size for sequencing. And then in the last step, what you sequence are the two ends of each fragment. So the earlier technology called single-end sequencing, you get to sequence both ends, but you don't pair them up. You lose the pairing information. You only keep individual ends. And the later, I think in 2008, 2009, 2010, the paired end sequencing allows you to keep the pairing information. So in other words, you get to know those two ends, I call two reads, are from one fragment. And this is more meaningful. So you can see that it's a very complicated experimental process. And what we get to analyze are the reads. And what we try to infer are something about the full-length mRNA. So you see, given all those steps in between, it becomes a statistical problem. And here in my talk, I focus on the reference-based RNA-seq data analysis. Because in many species, we don't have a reference genome. You don't have the DNA to compare your RNA-seq reads with. So you have to only use the reads to do assembly and to try to infer what are the possible transcripts. That's a different problem. But for species like human and mouse, we do have a well-annotated reference genome. So in that case, the current practice is that we first align the RNA-seq reads to a reference genome. In other words, we convert those reads into genome coordinates, and then we use those coordinates to do analysis. And this diagram just summarizes the three main levels of RNA-seq analysis currently. The gene level, if you take a look at that, what we care about is to infer the gene expression level using RNA-seq data. And among all the three levels, this is the simplest one. So after you have the alignment, you would know, okay, how many reads are aligned to a gene. And you count that read. So that will be here, I think this is out of power. so that count is denoted as N here. So then this is your gene expression level measurement. And we also have the exon level. And now in the exon level, our interest is often about the alternative splicing, which is about this blue exon here. So you can see that during alternative splicing, this exon is sometimes included in an mRNA, but sometimes excluded in an mRNA. So if we're interested in the ratio of those two types of mRNAs, then this is the question we're trying to infer. And this notation mathematically, actually, we should use PSI instead of phi. PSI stands for person spliced in, and that stands for the chance that this exon is included in mRNA. The most challenging level is the transcript level. In other words, we will try to infer the full-length transcripts from RNA-seq reads. And here the challenge is that some of the reads are ambiguous, which means that their transcript origin could be more than one. Like this green read, it could come from this transcript or this transcript. So in this case, how do you use those ambiguous reads to infer the expression levels of each transcript? That's a statistical question. So after summarizing those three main levels of RNA-seq analysis, which applies to both single-cell and bulk RNA-seq, I want to give you a very brief introduction about the contrast of single-cell and bulk RNA-seq. So the difference between single-cell and bulk RNA-seq is that in single-cell, you have the cell information recorded before you do the sequencing. In other words, now after you obtain the RNA-seq reads, you are able to assign those reads to individual cells. So you have more information. But on the other hand, you are still sequencing all the cells' RNAs together. So you didn't get much from sequencing, the sequencing depth. So you have to divide actually the total sequencing depth by the number of cells. So that means the signal you get per cell would actually decrease as your number of cells increases if you keep the total sequencing depth the same. So in other words, we don't have as many reads per cell as we have here in the bulk case. We have how many reads we have per tissue. So given the small number of reads per cell, for single-cell RNA-seq analysis, now we mostly focus on the gene level. In other words, we study the gene expression levels per cell instead of looking at the transcript level much, because that requires a lot more read information to study the full-length mRNA. So mathematically, for single-cell data, we have a matrix from a single-cell experiment. But for bulk, each sample is only recorded as a vector. Each entry is a gene, and the vector represents the tissue you sequence. So the first part of my talk will be about our recent work in the area called transcript or isoform quantification. The biological question we are interested in studying is the alternative splicing again. So given a gene, this is the DNA, it has 4 exons, and after alternative splicing, the mature RNA we obtain in this toy example will have 2 isoforms, like exon 1 to 4 and exon 1, 2, and 3, 4. You see that they differ in having exon 3 or not. And so mathematically, if we assume that the possible isoforms are combinations of exons, then given 4 exons, we will have a total of 2 to the 4th minus 1 possible isoforms, because every isoform should contain at least 1 exon. Then the first question you may wonder is, out of those possible isoforms, what exists in the biological sample, in the sample you sequence? And this problem is called as the isoform discovery or transcript assembly problem. And we already have multiple methods in the field, including the most popular cufflinks and the recent string tie and my own method slide all fall into this category. So in this case, we are trying to identify the existing transcripts or the expressed isoforms among the possible ones. So this is a very big area and it's still very challenging. And the second question is about our known isoforms, which means the isoforms we already discovered and recorded in annotations. So if we have a set of isoforms, say isoforms 1 and 2, that are annotated, biologists think they're reasonable, then what are their expression levels in the biological sample we sequence? This is the second question, and it's actually relatively easier because usually the number of known or annotated isoforms is not as large as the number of possible isoforms. So this question is referred to as isoform quantification or isoform abundance estimation. So I will use those 2 terms exchangeably in my talk. And this is my question of interest for this talk. So as another example showing this, as the same toy example here, we see that DNA, we have exons, mRNAs are exon combinations. And what we observe are the reads, which are those ones. So you see that in this case, the first read is called ambiguous because it's compatible with both isoforms 1 and 2, while the reads 2 and 3 are only compatible with isoform 2. So I have a clear assignment for reads 2 and 3, but not for 1. Oops. All right, so then for isoform abundance, we need to estimate that. Ideally, our goal should be to estimate the abundance of known isoforms and their copies. So let's say if we denote the copies of the first and second isoforms as N1 and N2, which are both non-negative integers, then our ideal parameter of interest will be the true proportions of those isoforms, which can be written as the Nj for the jth isoform divided by the sum of the total copies of all isoforms. This will be our ideal parameter of interest. However, we didn't get to sequence those full-length mRNAs in our second generation sequencing. So therefore, we don't have an observation of those copies, and therefore this quantity, the ideal parameter of interest, is not estimable. What is actually practically estimable is this probability that a random RNA-seq read is from isoform J. So in other words, because we observe the reads, Then using the reads we counted and we assign to isoform J, we can estimate this probability. And this practical parameter of interest in my talk will be referred to as the isoform abundance. So we put those probabilities of J isoforms together in the vector and denote this boldface alpha as the vector of our interest, isoform abundance. All right. So before we start the modeling, we need to have some notations to denote the isoforms we know. We want to estimate their abundance. So in our work, we define a subexon as a transcript region between adjacent splicing sites in any annotated mRNA isoforms. So in this case, if you have 3 isoforms, 1, 2, and 3 in my annotation, what we do is that we look for the nearest adjacent splicing sites and then divide this gene into those regions. Like each subexon is at least expressed or included in one isoform. So using this 1, 2, 3, 4 subexons, we can write out those isoforms just as sets of subexons. Isoform 1 becomes 1, 2, 4, isoform 2, 1, 2, 3, 4, and isoform 3, 2, 3, 4. This gives us convenience to denote isoforms. How about our data? How do we represent RNA-seq reads? You see here for the abundance estimation, the key information is the subexons that the 2 ends of a paired end read are in. So in this case, if we see the left end falls into subexon 1, the right end falls into subexon 2, then we will use this information denoted as 1 and 2 here. And also the relative position of those starting position and ending position of each end within the subexon may still be meaningful because we observe that the distribution of reads within the subexon is not always as we expected uniformly. If we want to account for such distribution, we will record those positions. So if we do it this way, then we actually record the full information about an aligned read. So then the question is, how do we infer isoform abundance given the reads? So here I just list 2 main categories of methods or models for this purpose, and both are based on likelihood inference. So the first type of models are called region-based models, and this is from the first paper that does isoform quantification back in 2009 by Zhang and Wang. So in this model, the data are actually summarized reads into region counts. So here, the region could be subexons or subexon junctions because some read may have the left end in one subexon, the right end in another subexon. So we have the count. And we have an index set for all the regions of interest. So then for a region in this index set, small x denotes the region. We use xs to denote the number of reads mapped to that region. And this is our data. And it's considered as the random variable here. So the basic model assumes that this count follows a Poisson distribution. And the Poisson mean parameter here is actually a weighted sum. Actually, it's actually a sum of this alpha j, which is the parameter of interest, the isoform abundance of isoform j. And ASJ here is the indicator indicating whether this region composed of subexon or subexon junction is compatible with isoform j. If yes, then it's 1, otherwise it's 0. So then you see, writing out this distribution, we can write out the likelihood, and the likelihood will involve alpha as the parameter of interest, and then you do maximum likelihood estimation to find the alpha. So although this can be a numerical problem, but the basic idea is like this. And later, people also can generalize this Poisson model to generalized Poisson model or negative binomial model, etc. So, but the key idea is to summarize reads into counts and then model the count distribution. And the second type of models is called read-based models. And we can imagine that for the first type model, once we summarize reads into region counts, we lose some of the reads information. For example, the relative position of a read within the region. The read-based model uses the full information from the data. And the idea here is that we regard each individual read, say Ri, as a random variable. And this is our total number of n reads. Then we write down the likelihood as a joint distribution of all the n reads. The parameter of interest remains to be alpha, and we assume the reads are independent. So we take this product, and then for each read Ri, we consider its probability from the isoform J times this parameter of interest, the probability of isoform J. And for this part, we call the generating probability of read I from isoform J, we can further decompose it into a product of the density of the starting position of read I and the density of the length of read I. And so you can see once we do this decomposition, we are actually assuming the starting position and the length are independent. This is for modeling convenience, but you may argue that we can just model them jointly. That's possible. And also, the bias correction about the read distribution is often incorporated into this SI density. Okay, so with this likelihood, we can also do maximum likelihood estimation. But here you see, because of the sum, each read can possibly come from all the J isoforms. This MLE is not easy. So that's why we can write a complete likelihood by introducing a latent variable about which isoform each read is from. After we introduce the latent variable, the complete likelihood will have a simpler form, and then we can apply the EM algorithm to find the maximizer of this likelihood. So this is the read-based model. And my talk today, our method will also be based on this read-based model. So this is just a very incomplete list of the methods that does isoform abundance estimation in the field, such as I mentioned, the first paper. It doesn't have a name, so I just give a short name here, the cufflinks, and RSEM, which can assign ambiguous reads, or actually the multi-mapped reads into isoforms, and my own method, which is actually more like a regression-based model I didn't introduce, but the idea is pretty similar. The express, which is actually after cufflinks, it's a modified version of cufflink that runs faster, and also other methods listed here. And even you can see, even until last year, we still have new methods coming out on this topic. So it's still a very active area of research, because given the data we lose so much information about the full-length isoform. So we are still trying to do better. All right. So here the motivation for my method, my talk here, is the existence of multiple RNA-seq samples for some commonly studied cell lines. For example, here, human embryonic stem cell is one of the most widely studied cell lines in labs. And we already have many samples in the public data repository, like the gene expression omnibus. Here, I'm just showing the RNA-seq sample, 6 RNA-seq sample about the cell line. If we focus on some of the regions here, you can see that in some regions, those 6 samples do not agree very well, but in some regions, they give you a very consistent signal. Okay, there's expression. So in this case, if we imagine that experiments can sometimes be contaminated, or some experiments are better done than some others, so then if we have the multiple sample information, a very natural question is, can we combine those multiple samples to do better about our inference of isoform abundance? So then how to combine the multiple RNA-seq samples is the key question we've tried to address here. Let's say that we have a total number of capital D RNA-seq replicate samples, technical or biological. If they're about the same cell line, then we regard them as biological samples because they intend to give you the same information. If they're not, then that may be something you don't care. And we have gene annotations about isoforms. Then the question is how to estimate the abundance of each annotated isoform for every gene. The very naive idea is that we just apply the existing method that works for a single RNA-seq sample to each sample separately, and then we average their estimated isoform abundance across those samples, right? We can do this approach. However, the shortcoming or limitation of this approach is that in the estimation step, you only use one sample and you didn't borrow information from other samples. So you didn't fully use the multi-sample information to reduce the variance in your estimation. The second idea is that how about I just simply pull those D samples into one sample and apply a method to the pool sample. This also sounds very reasonable, but the limitation here is that your estimated isoform abundance may be biased by some outlier samples. If you assume all your samples are good quality, then this sounds reasonable. But if some of your samples are actually poorly sequenced or contaminated, then if you do a pool sample, you will not have the ability to tell which samples are contaminated, and then the results may be biased. So there are some methods in the field that does multi-sample inference of isoform abundance. However, all those methods assume that the multiple samples are equally good, and they didn't consider the possible existence of outlier samples. So this gives us the opportunity to develop a new method, which we call short as MIS-IQ. It stands for multiple samples for isoform quantification. And the unique advantage of our method is the consideration of outlier samples. So first, I will introduce the notations for one sample, and then we will generalize it to multiple samples. Here in our model, the observed data are the RNA-seq reads. We also introduce latent variables as the hidden data about the isoform origins of reads. We don't observe this, but this will be part of our modeling process, and we will also infer those. And the parameters of our interest is the isoform abundance, again, alpha. So we are already familiar with those notations. And for D samples, we just introduced the superscript D to refer to the Dth sample. And here we have another layer of hidden information. That is, whether the Dth sample is an outlier or is not. If it is not, then we call this sample a consistent sample. So we introduce a binary indicator to indicate the consistency of the sample with the majority of samples. So this indicator is 1 or 0. 1 means the sample is good, 0 means it's not. And our parameter of interest is still the alpha. But here alpha means the common parameter of the consistent group of samples. So we allow each sample D to have its isoform abundance encoded as tau D. But tau D is either alpha if the sample is consistent, or is its own parameter beta D if the sample is inconsistent. So it's a combination of alpha and beta D. And this is the diagram of the whole model. It's called hierarchical modeling, or it also has the flavor of Bayesian modeling because we have the hyperdistributions. So here you see that for every sample, this part is the read generating process we already discussed about how we generate from isoforms. And this tau D is the isoform abundance of each sample we're interested in. And this tau D depends on the consistency indicator and also a distribution, Dirichlet lambda, to generate the isoform abundance. Dirichlet has the property that its observations from a Dirichlet distribution is a vector that sum up to 1, which is very nice. And this indicator, consistency indicator, we assume it to follow a Bernoulli distribution because it's binary. And for the Bernoulli parameter, which is between 0 and 1, the gamma, the probability parameter, we will assume it coming from a beta distribution. So that's the hierarchy of this modeling process. And it looks complicated, but it actually agrees with what we intend to do for this modeling. So as I said, tau D is a combination of alpha and beta D depending on the indicator. And both alpha and beta D come from Dirichlet. All right, so then we write down the joint distribution of all the variables in our model, the reads, the latent isoform origins, the isoform abundance, the consistency indicators, and this gamma is the probability that a sample is consistent. So they all together can be write out in their joint probability in terms of lambda, the Dirichlet parameter, A and B, the beta distribution parameter about this gamma. And we can decompose this long distribution formula into the product of 4 components. The first part is actually the joint probability of reads and their isoform origins, given our assumptions on the read generating mechanism, including the starting position distribution, the length distribution, etc. And the second orange part is the probability of isoform abundance given the Dirichlet and given the sample consistency indicators. The third part is the probability of the sample consistency indicators given the Bernoulli. And the last part is the probability of this gamma, the proportion of consistent samples given the beta. So our inference will be based on that. And to simplify our inference, because we know that our ultimate parameter of interest is alpha, and its inference becomes obvious given the latent isoform origin Z and the sample consistency indicator E. So we just integrate our gamma, the tau, so we just integrate a tau to make it simpler. And after do that, we use the MCMC approach to draw samples using the Monte Carlo approach to come up with the estimator. In here, because we assume nice distributions that are conjugate with each other, we can use the simpler form of MCMC, the Gibbs sampler, to draw samples. So what we do is that after we have this form, this distribution, we draw samples of Z, E, and gamma, because these three are not observable. But given condition on the fourth one, actually we have a nice distribution. We have a nice distribution for each of that. So given R, E, and gamma, Zi follows multinomial. Given R, Z, and gamma, ED follows Bernoulli. Given R, Z, and E, gamma follows beta. So this allows us to draw the samples using the Gibbs sampling approach easily. With those samples, we can have our estimator of alpha. So that's our final goal. All right, so this is our estimator for the isoform abundance. And those things are what we draw by Gibbs sampling. So here, using our estimator, we also define a measure, which is used in our paper, to quantify the relative estimation error between our estimator. So you can see it's actually defined as the absolute difference between our estimator and the true abundance, and divided by the true abundance. Of course, for real data, there's no way for us to calculate that because we don't have the true abundance. But in simulation, and we can investigate this. Another advantage of our method is the proposal of an estimator of sample consistency. So based on our Gibbs sample, we should be able to tell how many samples are consistent. Well, what proportion of samples are consistent? We can also assign a posterior probability to each sample D about its posterior probability of being in the consistent group. All right, so because of the time limit, I will just quickly show a comparison result based on some benchmark nanostring data. So the simulation results are in our paper. So in this nanostring count, we actually have 6 samples from a cell line, HPTG2. And for those samples, we have both nanostring counts and RNA-seq data. So there are 2 ways to convert nanostring counts into isoform abundance. I have to say neither is ideal or perfect, but it gives us some way to compare our estimates. So the interesting result is that you can see for the first 4 samples, our MSIQ estimate has better correlation with nanostring compared to competing methods, cufflinks, IRACA, and MISO, which is expected because we use their consistent information more. But on the other 2 samples, 5 and 6, we didn't do as well. And the reason is that because based on the judgment of our method, sample 5 and 6 looks more different from 1 to 4. So that's why they're kind of regarded as the outlier sample bar method. And that's why they didn't do as well as expected. All right, so the conclusion for this, I focus on the methodology. And the conclusions is that it's necessary to consider the heterogeneity of different samples to make robust isoform quantification. And method, MSIQ, is able to identify a consistent group of samples that are most representative of the biological condition. And MSIQ increases the accuracy of isoform quantification by incorporating information from the consistent multiple samples. And our hierarchical model is actually an umbrella framework that are generalizable to incorporate more delicate consideration of regenerative mechanisms. So if you have better ways to correct bias, etc., you can put them into this framework. Okay, and this is our paper published in the Annals of Applied Statistics this year and our R package. Any comments from the audience, if you're interested in this, is very welcome. The second part is the single cell, which I will quickly summarize because I think the idea is very simple and also I feel like the messages here should be generally applicable. So for single cell RNA-seq, what we focus on is the imputation of dropouts. So again, just to remind you, this is the single cell data, this is the bulk data. So as we increase the resolution to cells, we also lose the resolution per cell. So dropout refers to the case where you actually expect a gene to be expressed in a cell, but the observed expression level is 0. So in this example, we have two cells that are expected to be very similar. But if you look at the scatter plot of gene expression between the two cells, you see that a lot of genes are in the two diagonals, which indicate that the gene is moderately or highly expressed in one cell, but zero expressed in another cell, which doesn't make sense biologically. We may expect some of them, but not too many. And this is referred to as a dropout, which occurs when the transcript is expressed in the cell, but entirely undetected in its mRNA profile. Dropout events occur due to low amounts of mRNA in individual cells. And the two commonly used platforms for sequencing single-cell RNA-seq, including the Fluidigm platforms, which sequence hundreds of cells and get about 1 million reads per cell, or the more recent and more high-throughput droplet microfluidic platform, which can allow you to sequence like around 10K of cells per experiment. But the price you pay is that you get fewer reads per cell. So the trade-off is that given the same budget, which means the same sequencing cost and sequencing depth, you have more cells, then you have more dropouts. So for single-cell data analysis, then a lot of the focus are about the cell clustering. How do we cluster cells based on their single-cell data? To construct cell relationship, here is not a complete list, I'm just listing Sura, but there are also like Monaco and other methods which try to find the pseudo-time of cells by ordering cells given their expression profiles, and dimension reduction techniques which try to find the low dimensional representation of cells so you don't have to look at the expression levels of thousands of genes per cell. You can use this low dimensional representation to better see the cells. Okay, so but imputation I think is still very useful, although those methods all implicitly consider the dropout issue in their modeling for their specific purpose. But we think for biologists who use single-cell data, they usually do not have the expertise to manually do the dropout consideration in their specific analysis task. So therefore, to make it easier for biologists to analyze the data, it's better to do the imputation for them, and then they can use the imputed data to get better analysis results. So that's why we think genome-wide explicit imputation is definitely needed in the field. And there are methods that does genome-wide imputation. The first method, MAGIC, was developed, I think it was put on arXiv in 2017. It's the first method for explicit genome-wide imputation of single-cell RNA sequencing data, and it pins missing expression values by sharing information across similar cells. This approach is to create a Markov transition matrix which determines the weights of the cells. So when we first read the paper, our question is, why is a Markov transition matrix applicable? So what is the Markov chain here? So by reading the paper, we understand that their argument is they want to find the differentiation trajectory of cells. From that perspective, you may expect a transition across cells. But in many cases, if we just sequence cells that do not follow a differentiation trajectory, then the Markov chain doesn't have a very intuitive understanding. And after we've posted our method to bioRxiv, there are two other methods posted to bioRxiv, the Saver and Dr. Impute. So please note that the MAGIC and Saver was just published in June. Dr. Impute is still on bioRxiv. So Saver uses a Bayesian approach, and it aims to estimate the expression of every gene in every cell as a parameter. So you can imagine it has many parameters, but you use a Bayesian approach to infer the prior parameters and then do the posterior inference, etc. And the Dr. Impute actually uses the averaging of multiple imputation results. And also, there are several other recent methods upcoming on bioRxiv. So we can expect that this area is also very active. All right, so the motivation for our method is that we first argue it is not ideal to impute all gene expression because some of the zero expression could be biologically true. And also, if we are too ambitious, if we want to just alter every gene expression value, we may actually introduce new bias from our statistical modeling. And the second argument of us is it is inappropriate to treat all the zero expressions missing values. I just said that. So based on those reasons, we think it is very important to to use a step to discern which values need imputation and which may not need. Okay, and how do we do that? So our method called SC-impute has two main ideas. The first idea is that for every gene, we want to determine which of its expression values are most likely affected by dropouts. In other words, the values need imputation. Second, for each cell, we only want to impute the expression values that are highly likely dropout, and we only borrow information from the same gene's expression in similar cells. So in other words, we take a very conservative approach and we don't assume a similarity relationship between genes because we think the gene-gene relationship is dynamic. It may change from cell to cell. So it's safer to not use that information, but instead we think it is natural to think the cells fall into subpopulations or subtypes, and within each subpopulation the cells are similar. So it's safer to borrow information within the subpopulation. And this diagram demonstrates our idea. So you can see for every cell, we use the first idea to discern, like those need imputation, those do not. And then we use those do not, called gene set BJ, to identify the cells similar to cell J. And using those identified similar cells, we use the gene expression in those similar cells to impute the values of the genes in set AJ. So that's our idea. All right. So data preprocessing, I will just briefly say that we use the log transform gene expression because after we do the log transformation, we convert the gene expression from counts to continuous values. That makes our modeling more easier. And so the first step is to identify cell subpopulations and outlier cells. I think this is a unique step of our method because many other methods didn't have this step. And what we do is that we first do dimensional reduction, and then we do a cell-cell distance matrix called D. Based on this distance matrix, we detect outlier cells. And then we cluster the remaining not outlier cells into subpopulations by spectral clustering on this distance matrix. So you may argue that this is a parameter to tune, but in practice, often biologists have a brief idea about how many subpopulations are in the sample. So they have an idea about what the range of this K. All right, and the second step, what we do is that for every gene, we model its expression as a mixture of gamma and normal distribution. So here the gamma component represents the dropout close to 0. Normal represents the actual expression. So with this, I will show you a figure to see why this is reasonable. With this, then for every gene I in cell J, we can have a posterior probability that this gene in cell J may need dropout. So this is the posterior probability of dropout. And here we can see that the two parameters that need attention, the first one is the estimated dropout rate lambda I hat K in subpopulation K. So this only depends on gene I, but not on cells. This is the overall dropout rate of gene I. This Dij hat is the estimated dropout probability. It depends on both the genes and cell. And we will use the Dij hat to help us decide which values need imputation. So this is a way to show that the gamma Gaussian mixture makes sense. So the blue bars are the histogram of actual data, and the solid black curves are our estimated distribution. So you can see that they fit pretty well. And then based on our Dij hat, we will apply a threshold T to actually divide the genes in the cell into set AJ that need imputation and BJ that doesn't need imputation. So regarding the choice of T, we want to say that it's not very difficult because if you look at the distribution of Dij, you can see that most of those are very close to 0 or very close to 1. So if we set T to be 0.5 or 0.4 or 0.6, our decision on AJ and BJ will not be too different. All right, and then with the AJ and BJ, what we do is that for each cell J, we use gene set BJ and the non-negative least square, which naturally introduce sparsity to learn which cells in the neighborhood of cell J or in the same subpopulation of cell J are similar to cell J. So this does the cell selection process and also estimate the parameter, which are the weights of the selected cells, using this non-negative least square. And then we use the estimated coefficient to infer the gene as the expression in set AJ as follows. So then you can see if the gene I is in BJ, we don't do imputation. If the gene I is in set AJ, we do imputation using the estimated coefficient from this step. So this is a summary of our method. We think the advantage of our method compared to other methods in the field is that it's very interpretable, and also the steps are decomposable. So you can really understand what's going on in each step. And if you have better ways to modify each step, it's easy. So the first real-data example is just to show with the ERCC spiking, which are used as the sort of like the gold standard for validating single-cell experiments. So the ERCC transcripts has known concentrations, and if you look at the original data, you see that there are some transcripts with non-zero ERCC concentration, but their count in single-cell data is 0. But after our imputation, they are no longer 0. That's the first example. And the second example is to use the very famous PBMC data set in the single-cell field. It has 4,500 cells from the 10x Genomics company's technology. And this is the clustering result based on original data, and this is based on our imputed data. So you can see now the clusters are better separated using the tool called T-SNE for two-dimensional visualization. And you can see it's better separated. And third, about the gene expression dynamics. Here we use a very valuable data set that has time course single RNA-seq data, both bulk and single cell. And so you can see that for some time points, we have 3 replicates of bulk, and we have hundreds of cells of single cell. So the idea we want to use this data is because we think on average, single cell, if we take the average across cells, it should look like bulk, and we want to show that. So you see that after imputation, the correlation between single cell and bulk did increase, which is expected and very nice. And also, if we look at some genes that has been found as related to the differentiation. So you can see here the black marks are the bulk expression, and the box plot shows the single cell expression across cells at that time point. Before imputation, their agreement is not very good, but after imputation, their agreement becomes much better. So just to summarize this part, the method SC-impute is a flexible and easily interpretable statistical method that addresses the dropout event prevalent in scRNA-seq data. And it focuses on imputing the misexpression value of dropout genes while retaining expression levels of genes that are likely unaffected by dropout events. It is also compatible with existing pipelines or downstream analysis of scRNA-seq data, such as normalization, which should be applied before we do the imputation, differential expression analysis, clustering, and classification. And also, SC-impute scales up well when the number of cells increases. All right, so this is the paper and our software. If you are interested in it, please try it, and also your feedbacks will be very appreciated. So finally, I want to thank my PhD student, Vivian, who is also a volunteer here, and she's the main contributor to both projects, which I think is very impressive during her PhD study, and also our funding agencies. And also we are looking for postdocs. If you are interested in our research, please check our website and send us emails. Thank you very much.
Probably have time for 1 or 2 questions.
I don't know, maybe I missed it, but do you focus on just a certain set of genes?
You mean for the second part, or
Yes, for the second part.
We actually so this is a genome-wide imputation. We didn't choose a gene to work with. So basically, if your data set contains 4,000 genes, we will use all those 4,000 genes. But of course, we will remove the genes that are 0 expressed in every cell.
But for example, if you look at long-long so most likely you have very few of them because they are so lowly expressed, but also you don't really know if they are even consistent between different cells because we don't know if they should be expressed in every cell.
That's right. So that's why I think the key step of our method is this step. So we will actually, for every gene, we will look at its expression values across the cells in one subpopulation. So if the gene has a bimodal distribution, then we think it is likely drop out in the lower mode, in the cells in the lower mode. If it's a unimode distribution, whether it's close to 0 or whether it's highly expressed, then we think that gene may not need dropout.
So you don't discriminate between genes. You can also look at microRNA.
Correct. That's right. That's right. Yeah.
Any other questions? So let's thank Jessica again.
Thank you.